Hello, I'm Deborah Davidson, Director and Founder of Catalyst Conversations, and I'm so happy to welcome you to a Catalyst Conversation. I want to thank the Broad Institute for its continued enthusiasm for Catalyst, and to Shannon Humphreys in particular for working together to make the program possible this afternoon. Catalyst Conversations was founded to open a critical path for dialogue between the arts and sciences. We present intimate and provocative conversations between artists, scientists, and the public. We're interested in connecting the two through programs, educational outreach, and public events like this, which demonstrate the important conversation and synergy between art, science, and technology. This afternoon's topic is Women in Art and Science, the Criticality of Hope. The theme of the conversation is inspired by Bina Venkataraman's recent book, The Optimist Telescope, Thinking Ahead in a Reckless Age. The speakers will explore the criticality of hope and optimism in our culture and how practitioners in both the arts and sciences rely on hope in order to be forward-thinking, especially in this current moment. These women represent a broad range of both art and science practices. The program will begin with short introductions by each of our speakers. They then will share with each other their respective thoughts on the relationship between art and science, how and if their gender has influenced their performance and thinking, and how that affects their respective projects. A few notes. This program is a rescheduled version of what we had planned for last March 19th. Maggie Stark and Michelle Samour are exhibiting their work at the Broad in the exhibition Worlds of Marvel. We welcome your questions and comments, and you can do so using the Q&A button to type in your question, and Shannon Humphreys will read them to our speakers. I will now introduce everyone. Bina Venkataraman is an American journalist, author, and policy expert. She is currently the editorial page editor of the Boston Globe and a fellow at New America. She is the author of The Optimist Telescope, Thinking Ahead in a Reckless Age. A former advisor in the Obama White House, she helped communities and businesses prepare for climate change, and she learned firsthand why people don't think ahead and what can be done to change that. In The Optimist Telescope, she draws from stories she has reported around the world and new research in biology, psychology, and economics to explain how we make decisions that benefit us over time. Michelle Samour is a multimedia artist whose installations, drawings, and handmade paperworks explore the intersections between science, technology, and the natural world. She has exhibited at the Decorative Sculpture Park and Museum, Museum of Modern Art Strasbourg, Kohler Art Center, and the Houston Center for Contemporary Craft, and the Fitchburg Art Museum. She has received grants from the Mass Cultural Council, the Society of Arts and Crafts, New England Artist Award, and grants from the Cushman Family Fund, Andrew W. Mellon Foundation, and the Dana-Farber Fund. Maggie Stark is a multimedia artist whose videos, photographs, and sculptures explore the nature of time within real and imagined spaces. She has exhibited widely, including solo exhibitions at the Caiaphas and Hull Space Galleries in Boston, the Burroughs Rotunda, Dartmouth College, and McKinnon Art Gallery, Southern New Hampshire College. She has held residencies at the Corning Museum of Glass, Malay Colony and Vermont Studio Center. Recent awards include a cultural fellowship from the Goethe Institute in Berlin and an artist residency fellowship at the Halsa Art World Museum, South Korea. Cigall Kadoch is an institute member of the Broad Institute and an assistant professor of pediatric oncology at the Dana-Farber Cancer Institute and Harvard Medical School. She studies chromatin regulation with a strong focus on structure and function of the mammalian BAF family of chromatin remodeling complexes in human cancer. Her work has been centered on interrogating rare, molecularly well-defined cancers to understand the role these complexes play in promoting a wide range of more common types. So with that, we'll start with Bina.
Thank you, Deborah, and it's a pleasure to be here, and I'm honored to be in this group of people. And as you heard from my bio, I am neither an artist or a scientist, though I take my inspiration from both as a writer, as a policy thinker, as someone trying to really drive social and political change. And of course, climate change has been one of the animating reasons and sort of motivations behind my work, including behind the Optimist Telescope, but it's not limited to that. And I I guess the first thing I feel is important to say is that I am not a person who urges everyone to be an optimist or to look at the world through rose-colored lenses. That is not the kind of optimist I am. That is not what the Optimist Telescope is really about. It's actually about being able to imagine different kinds of futures, being able to act for the sake of the future. And I think of that as a form of engaged optimism. It is not believing that the world is inevitably and inexorably driving towards progress and getting better. It's believing that the future is changeable. And so that, I think, is an important distinction. And I will just say two things about what is missing, what often gets in our way of being able to see the future as changeable, and the role that I see both science and art playing in that. And I hope that will be a little bit of a spark for the conversation. And then we're going to hear from the artist and the scientist on this panel. And I think we'll have a rich conversation based on that. But the first is really that there's an imagination gap. So if you look at research from people across countries of the world, we find that most people's imaginations of the future go dark after about 15 years. Increasingly in our lives today, it's not even the case that we think much farther than the next day, the next minute, the next moment we're going to get a reply to our email thread or our text thread. It's very easy for us to stay locked on the immediate. And our ability to actually imagine the future looking different than it does today. I think in particular, yesterday we heard some positive news, promising news about a potential COVID-19 vaccine. And I think it shifted some people's ability to think about and imagine a future after this pandemic. But I would say up until yesterday, even for me, as someone who considers myself a person who cultivates imagining the future, that living in the pandemic, it's been very difficult to kind of look at and think about what the future looks like. What does it look like to be able to travel again, to be able to see our families again, to be able to go and interact with people the way we used to? And what does it look like to not live in fear of this virus? Well, part of the problem when we're living through crisis is that we lack the ability to imagine that change can be nonlinear, that there can be something as dramatic as if there is a vaccine that is effective, that's actually distributed widely in the population, it can dramatically change our reality. I think the same is true of political crisis or political polarization. Until that moment of inflection in political change or social change happens, whether it's an election, whether it's a successful civil rights movement, as we saw in the 60s, until that moment happens, it's difficult for a lot of people to be able to imagine it looking different. But I think that progress actually hinges on people being able to have that imagination. And art and science have a role to play in this ability to imagine futures that are very different than the present. So whether it's John F. Kennedy drawing on the inspiration of scientists and engineers to say he envisioned people being put on the moon and then driving all that scientific progress towards that imagined future, or whether it's scientists today who are dreaming about what kind of fuels of the future will look like, what kind of medical interventions of the future will look like. That imagination is so critical to being able to actually drive that progress. It doesn't mean we'll always arrive at the imagined future, but it does motivate people, and it does allow people to unlock themselves from the bias of the present, the constraints of the present. Similarly, artists, I write about artists in my book. For example, I write about a project set up at a cathedral in Germany to play a piece of John Cage music, As Slow as Possible is the name of the piece of music. And the organ that is being used to play this piece of music is being assembled and reassembled and disassembled over time. And the piece of music is actually going to be played over the course of more than 6 centuries. And so every several years, a different note or combination of notes sounds on this organ. And you might think that sounds sort of esoteric or strange, but one of the reasons I wrote about projects like this that come from the arts, that come from artists, being able to think across time horizons, is that it allows us to then have an anchor. If you start to think about this piece of music being played 600 years in the future, does that allow you then to open up the space to think about what the world looks like in that future, what the people look like across that time horizon? So imagination is really critical. The other piece is to be able to think about beyond short-term and immediate distractions, short-term and immediate gratification. And here I think both science and art have a lot to teach us as well. Art has the ability to transcend the moment that we are in. It has the ability, when you're standing in front of a painting in a museum or gallery, when you're standing and watching a piece of performance art, to be able to leave the moment that you're in and inhabit the world that the artist has created for you. Similarly, I think the history of scientific progress and the present of it is an important, it offers important lessons for us about how the process of going slow, the long-term payoffs of research. Sometimes research happens over the course of decades before it transforms the world and transforms our lives or yields insights. And so I think both of these fields, for me, offer a way to unlock us from that very present focus that we have, and the distraction that we have, and our tendency to measure ourselves by the immediate. So I'll leave it at that, because I really want to hear from the other panelists and really want to be part of the conversation. But I hope that provides a little bit of my thinking about what I really mean by optimism and what it means to really imagine the future.
Thank you, Bina. Michelle.
So thank you, Deborah, and Shannon, Catalyst Conversations, and the Broad. It's an honor to be with all of you today. As Deborah said, my work explores the intersections of science, technology, and the natural world, whether it's the tentacles of a jellyfish, fiber optic cables, the flagella of microorganisms, the symbiotic relationship of mycorrhizae and plant roots, or abaca plant fiber that I use in papermaking. I think about these associations in the broader context of collecting and cataloging. In this piece, Reflecting Pool Beautiful Viruses, dozens of translucent paper viruses, including SARS, smallpox, pox, Ebola, avian flu, and HIV, flowed into two-layered light box, the pool's rectangular configuration suggesting a microscopic slide, the virus's flower-like configuration suggesting an aquatic garden. Reflecting Pool Beautiful Viruses is a place to quietly contemplate and reflect on the potential of these seemingly beautiful viruses to do enormous harm. This piece is from my 2010 show, Truth and Transience, and sadly, I now have to update it with the coronavirus. The images for the next series of works begin with the basic non-objective drawing mirrored along its central axis. Variations on these forms are repeated in different ways and reference the Victorian era's embrace of nature, curiosity, discovery, observation, and excess, while also conjuring 21st century connections to genetics, cloning, and digital manipulation. It's a detail. In the Victorian age, the parlor was the primary gathering place in the Victorian home. Family and guests convened in the parlor to play games, talk, and play music. Specimens of butterflies, iridescent beetles, shells, and taxidermy birds were decoratively displayed under bell jars in freestanding fireplace screens and under glass tabletops. They were often a catalyst for educational conversations. And today, our conversations are often between our computers and ourselves. The digital display is stand-in for actual specimens and sometimes for humans themselves. Mm. So the effects of climate change on plant life is the focus of my most recent work. This piece, Blue, is a reflection or reinterpretation of the algae used by botanist and photographer Anna Atkins from her book, Photographs of British Algae, taking them out of her historicized book and bringing them into a contemporary context. Using paper pulp that I've pigmented blue, I'm referencing Atkins' original blue cyanotypes. The addition of reds and oranges suggests how the warming of the oceans and subsequent red tides or red seaweed have altered algae since Atkins recorded her specimens over 150 years ago. The resulting specimens I've pinned to the wall with specimen pins, and they reside between the actual algae that Atkins used and the final cyanotype photograph. So orchids have a long history as sensual and sexual plants. The early spider orchids have been dependent on bees to pollinate them, but global warming has resulted in this relationship being out of sync. So Cinque, early spider orchids, addresses the sensual and sexual relationship between the bee and the orchid flower, resulting in hybrids of the two. The format that I'm using is derived from late 1900s natural history educational charts that were designed to be seen from the back of the classroom. And finally, this series adaptation that I began in the summer of 2018 when I was fortunate to be a scholar in residence at the Tufts European Center in Talloires, France. The drawings are based on direct observation of the exterior and interior structures of the plants of the Haute-Savoie region, as well as my visual interpretations of how these plants are changing and have the potential for extinction due to global warming. Drawn with a quill pen, they reference early natural history illustration. In these hybrids, the organic is overlaid and interchangeable with the technological, and address some of the following questions. What are the visual and metaphorical overlays between optical and copper wires used in digital communication, and the function of the root hairs of a plant? And what does the function of the bee to the flower and the human to digital transmission have in common in regards to regeneration and multiplicity? And finally, how does global warming cause plants and animals to adapt? And what does that look like? So finally, I'd just like to share with you one of my favorite quotes from the great polymath, naturalist, and explorer Alexander von Humboldt. I shall try to find out how the forces of nature interact upon one another and how the geographic environment influences plant and animal life. In other words, I must find out about the unity of nature. Thank you.
Okay, great. I'm very pleased to be here, and thank you, Deborah, and thank you, Broad Institute. What a fantastic group of people, and so I'm pleased to be a part of this discussion. I'm just going to share my screen with you. Okay. The paradox of time, simultaneously perceived as finite and material, and infinite and permeable, provides a conceptual framework for my work in video, sculpture, and photography. Within this framework, I'm exploring such divisions as motion and stasis, inside and outside, you and me. Integral to these works are light, sound, movement, and repetition. The first two slides are from a series of photograms I created in an analog darkroom. I placed structures constructed from scientific glassware, glass rods, and fiberglass underneath an enlarger. As the light passed through these constructions, the resulting shadows were recorded and fixed onto light-sensitive paper. The measuring of incremental time, from exposing the paper to light, to the timing of the chemical baths, to the final washing of the print, afforded me a heightened awareness of time, while keeping at bay the actual passing of time. I was aware of the duration of seconds and minutes without comprehending their cumulative scope. I was caught in an eternal present. The next several videos, ending with an abbreviated video clip, are from my piece, Wall Play. I used the childhood game of hopscotch to examine the fundamental dualities of you, me, here, there. Unlike the game hopscotch, which produces an oppositional winner and loser, Wall Play's players, adversaries, are locked in a joint effort to illuminate and extinguish pre-existing patterns. The interdependence of these players' actions determines the resulting patterns of light and dark. Okay, this slide and the next brief video excerpt are taken from the video Time Lock. Time Lock, like wall play, depicts two figures engaged in a joint effort. Enclosed in cage-like structures that dictate their movements, each tries to chart her way around the other. The performers improvise to the sound of a metronome as waves of fog roll in and out, the arc of their actions locked in an abstracted nocturnal landscape, unstuck in time. Okay, the last couple of slides depict Loop, a praxinoscope, an animation machine with roots in the 19th century. The images on the interior wall are animated when the viewer spins the scope and stop when the viewer halts the motion. The imagery remains constant while its motion is variable, raising questions of whether we are moored in time, floating in time, or experiencing both in parallel.
Thank you, Maggie.
Yeah, thank you.
Cigall.
It's really great to be here, and gosh, that's such cool art and such amazing animations. I'm really taken by those. So thank you for the opportunity to participate. You know, I'm Cigall, and I'm a scientist. Our laboratory is really centered on understanding the structure and function of proteins that sit along our genome and actually regulate genomic architecture. And so really I'm thinking a lot about this quote that was just mentioned from Alexander Humboldt about how understanding the interactions, nature's interactions, and how critical that is to understanding the unity of nature as a whole. And so that's really, in essence, as I was just reflecting on that quote, that's in essence really what our lab is seeking to do, trying to understand how factors inside the nucleus of each one of our trillions of cells work together to coordinate all of our human functions, and how those functions can go awry in human disease specifically. So as you can imagine, our DNA contains all of our genes. And if you were to take the DNA out of any one of our given cells and take it out of a cell, which you can't even see by the naked eye, take it out of that cell, the DNA that you have would actually stretch 2 meters in length. That's a lot of genetic material, and that codes for all of life. And that same stretch of DNA is essentially present in every single one of our cells. So how must then a 2-meter stretch of DNA fit inside the nucleus of a cell that's less than the size of a pinhead, that's completely invisible to the naked eye? Well, it turns out there's a lot of factors that help this long stretch of our code, our DNA genetic code, fit inside the nucleus. And so there are proteins around which that DNA can spool. And so it's like a telephone cord progressively spooling that allows all of our genetic information to become compacted and inside the nucleus. Now, as a parallel, just like a box of books, if you were moving and you were packing up your entire set of books, much like what Bina has behind her, and putting all of that inside a box, and suddenly you really wanted to access that book at the very, very bottom of the box, you'd have to do some unpacking. So there are factors, it turns out, inside the nucleus of our cells that actually are the unpackers, if you will. They unpack very densely, tightly packed areas of our DNA, which is also another word called chromatin, which is proteins and DNA together. They unpack those areas so that one can access that book, or in the case of our cells, so that the cell can actually access and turn on a certain gene. Now, the criticality of turning on genes and turning off genes at the right times is important and instrumental in every single one of our human body processes. And it's actually the single main cause of human disease. It's aberrant gene expression. That is, which genes within a program of 20,000 genes are turned on and turned off at the right and wrong times. All of that explains cells either going right in the right directions and performing the correct functions, or going awry and performing incorrect functions, such as becoming cancerous cells, becoming neurons that don't function anymore, et cetera, et cetera. And so our lab is intently focused on understanding the factors that can control this sort of opening or unpacking of books and closing of the boxes full of books. And it's actually another analogy that was brought up is just an orchestra or an organ that has maybe been pieced together multiple times and put together and creating for different sounds throughout its lifetime. And similarly, cells have the same thing. An organ, for example, you can play a set of sounds, and those set of notes need to be played in concert and in harmony with one another. Just removing one note, for example, will produce a very different sound, or adding a note will produce a very, very different sound. And similarly, if you just think about that 2-meter stretch of DNA that I described, where these factors that our lab studies called chromatin remodeling complexes, or essentially DNA remodeling complexes, they just roll along the DNA and tell which segments of DNA to be nice and open, ready for action, ready to turn on the genes, and which sets of DNA to keep nice and closed. So those factors, it turns out, are major determinants of human disease. That's why we care about them so much. And in efforts that were spearheaded in large part by the Broad, these efforts identified that actually mutations in these factors are present in over 20% of human cancers. They're actually the driving hallmark pathognomonic main feature of several cancer types, including rare cancers, and also several autism spectrum-like disorders, intellectual development disorders, so several neurodevelopmental disorders. The single cause of many of those is a problem in just one piece of this large machine or set of proteins that we study that, again, rolls along DNA and tells the DNA which genes to make open and accessible and which genes to keep closed. So this is a major process underwriting essentially our entire existence. It enables every single one of our cells to perform its proper functions. And when it goes awry, we're learning more and more it's often because of a problem in one of these genes. And so our lab is, taking a collection of approaches. We try to integrate, and again, understanding the interactions of nature. We also understand the interactions of technologies and looking toward the future. As Bina mentioned, it's very, her definition, in part of optimism is being able to have that creativity and think beyond the immediate and think of the future. And similarly, as scientists, we've had to do that quite often, think about what are all the skills we have now, but what are those skills going to turn into, and what might we envision for the future in terms of how we understand this process, how much it impacts human function, human disease, and being able to have a constant level of foresight in terms of thinking about all this collection of skills and collection of tools and ways of thinking and conceptual advances we might apply to advance our understanding of this very critical area of biology. So thank you.
Thank you. I think we should all get unmuted. Everyone but, well, we're good. So when everyone's unmuted, we can start our conversation. And I just want to say how great all your short presentations were. I feel very kind of inspired, and it's just great to be with people that are passionate and thinking and caring. Bina, can you hear me? You need to be on. Okay.
I can. I will confess there is a tree whose branches are being felled right now. So I'm just trying to make sure I don't distract you all. As I was thinking of, and thinking about the humble quote and looking at the beautiful images. Yeah. There was also a bit of life destruction happening, or at least trimming. It might actually be life-saving for other plants.
Well, I think, well, I'll just explain. I'm going to moderate this next section, and I'm happy to go, not answer all my questions. I'm happy to just let things happen as they do. I mean, you've already, especially Bina, you've answered some of the things that I was going to ask. So anyway, the way I'd like to start actually is with a quote from Alan Lightman, the novelist and physicist, who said to me quite a long time ago, actually when I was just starting Catalyst, I had the pleasure of meeting with him for an hour at his house while I explained this idea I had, and he gave me his blessing. So anyway, he said that both art and science need metaphor to find something new. At some point, science needs to go beyond metaphor, and art can stay within that metaphor. So I think both Cigall and Bina have sort of touched on that, but do you want to, I don't know, anyone want to respond? If that does resonate with you.
Sure. Bina, would you like to start or want me to start?
Please go ahead. Yeah.
Sure. So, I think this is a very interesting parallel. We use in science all the time metaphors. In fact, I think I always look at the ability to make a metaphor often comes, at least for me, from my understanding or feeling a confidence that I'm understanding one aspect of the science in a way that's clear. The more we can make parallels with simple objects, simple processes that everyone understands, the more we've probably understood about a certain process. When I'm not able to do that, I think it more indicates to me that we have a lot more to dig up, a lot more to learn. And so there's a constant toggle, I would say, between scientific facts and new metaphors that we create in the lab. And I think in terms of going beyond metaphors, science really requires a proof of a metaphor. Right. And so we're often, bringing a metaphor to the table. My lab mates and collaborators, we're often bringing a metaphor to the table and then putting it on that table and leaving it there until it's tested. And people go off and go back to their benches to try to test that metaphor and see whether or not it truly applies, or maybe it applies in a subset of situations, but not in all situations. And that's something that I think we do every day. If I just think of the last 3 Zooms I had before this meeting and the next 3 that I'm going to have after this, I would say that's oftentimes a central theme of what we do. The science really, will need the proof of the metaphor, but the metaphor can merge, morph, change, evolve over time. And I think that's one of the beauties of science. And certainly that's true of art as well. Mm-hmm.
Yeah, it's, it's an interesting way to think about metaphor. So metaphor helps us understand things that are impossible to, that we are out of our grasp. And the artist can kind of keep doing that. And the scientist must kind of repeat, the hypothesis must be proved so that you can, create the drug and so on. So anyway,
right.
It's something that has stayed with me. And anyway.
It's a great quote.
I really love what Cigall just said, and it strikes me that I work in both of the modality of the scientist and the artist with metaphors. So there are times when there's a complex concept that I'm trying to encapsulate for people to help people understand. And then there are times where I'm just drawn to something for its aesthetic qualities or to describe it as a writer, and then find it later becomes a metaphor. And for this book, The Optimist Telescope, sort of a dominant metaphor in the book is the metaphor of the heirloom. And before I even started writing the book or had even conceptualized what the book was about, that it was about long-term thinking or what exactly my angle or my title was going to be, or my arguments or stories or research, I had received this heirloom from my great-grandfather, which was this instrument called a dhoruba. I had inherited this instrument, and I started writing about it and thinking about it, and it started to kind of draw me, and it became some of the early writing of the book, but I didn't realize what it meant and how it connected to the book. And later, in trying to extrapolate sort of this concept of how do we leave things to future generations when we don't know what future generations will live like? We don't know what discoveries Cigall's lab is going to make that are going to change their lives, how their health. We don't know how technology or the remaking of the world in different ways might change what it's like to be a human 50 years from now, 100 years from now. But we can understand and know what it's like to pass things between generations, right? To shepherd something because we know it was valuable to the past. we bring it forward, it has meaning in our lives, and then we pass it on to the next generation. And so this heirloom of the instrument, in a non-intentional way, in a very indirect way that I think of more as the way of an artist, later became a very much more explanatory metaphor, where I actually tried to tell the reader that I believe we need these kind of heirlooms, not just in our families, but in our communities, and we need them to leave them collectively as a society. And the way to do this is not to try to imagine everything about those future generations, but to protect what has been mattered the most to us, whether that's clean air or clean water or the forests that we rely on.
Yeah, that's beautifully put. Here's another question that maybe one of the artists would like to respond to, which is, how are you responding to the current moment, both in your work and personally? And Bina, when in her introduction, kind of touched on this. Maggie, do you want to?
Well, I brought up the being caught in the eternal present, and so not being forward thinking at this point, trying to understand what it means to be living now. And so I slowed way down and started thinking about the 366 days that are in this year. And I've always been fascinated with the change of light and dark, and how in the middle of the summer solstice, you have much more daylight, and then you go to darkness. And so I ended up taking the days and making them into cubes and painting each day to the amount of light that would be on that particular day. So I had these gradations, but the process of doing that was very incremental. And so I was very aware of the action of doing it. It was sort of almost like when I was in the dark room, and I was so aware of time. And so I just slowed myself down into the time that we're living in. And so that's one way I've been.
That's really interesting. I'm imagining what those look like. Are you going to show those at the show at Hall Space?
Yes, which is coming up in a couple of weeks. So
you'll let us know about it, okay?
Yes,
yeah,
I will.
Anyway, that's interesting how the thing that it is important to you, like, it has a different format in this moment. But I'm just thinking about the works that you showed us. You're interested in time, you're interested in light and dark, yeah. Michelle, do you want to jump in?
Yeah, just a thought too. I mean, I've been slowing down and focusing as well. So Maggie, and I think that just being able to be in the studio, to have that opportunity just to stay focused, but at the same time, my attachment to nature has increased that much more. A lot of us talk about how important it is to be outside, to walk. I think in general, people's attachment to nature has intensified. And that has really helped me with my work and has also allowed me a certain freedom to stay within that happy place, that bubble of nature. So instead of worrying constantly about climate change and how the environment is changing, I'm appreciating it more and staying within that sort of thought process, and that has helped me. So my outlook toward the future has changed, I think.
Can I just respond to what Maggie and Michelle just said, because I found both of those comments really profound, and it reminds me that thinking about this eternal present, and just when I was looking at some of the pieces when you were saying that phrase eternal present, Maggie, it made me realize remember that when I was researching the Optimist telescope, coming across this distinction that Isaac Newton had made, of course, famous scientist in our history, between mathematical time and relative time, so
Right.
time as we experience it. And it became so important to me to understand that distinction, because I'm also a person, despite being this sort of I guess, preacher of the practice of long-term thinking and imagining the future, evangelist for that, a person who believes in relishing the moment, as Michelle and Maggie have talked about. And I think it's in those moments of joy and connection to nature that people actually forge the ability to have concern and care to protect that heirloom for the future. So I think it's critically important. And there was this conundrum in my mind as I was writing about long-term thinking. How do you reconcile this eternal present, which has this gorgeousness that Maggie captures in her work, with this idea of needing to imagine the future and do things on behalf of the future, like Cigall is doing in her lab and trying to gesture towards in her work, which doesn't always have that immediate reward. And I think it's actually more compatible. I think part of what this kind of art and this kind of work does, as I look at Maggie and Michelle's work, is take us out of not that deep, timeless connection to the future, but that obsession with, sorry, I'm losing my, my headphones here, that obsession with the next immediate moment, and that compression of relative time, where each moment feels so short and it's gone, and it's just, we're always fixated on what's immediately next. And I think that that's the place that's full of anxiety. That's the place where we're blinded to both the future and the present moment. So.
And I think adding to that too, as a scientist, I think as I constantly remind, my trainees and remind myself, really, we're trained to deal with exactly this, in the sense that scientists are constantly revising expectations, changing course, having to adapt to new knowledge, new technologies, new situations, throw out a hypothesis, take a new hypothesis, take a completely different route toward getting toward answers. And I think that we're practiced for exactly what happened here. And so as we think about how have we revised our strategy during these past several months to still be able to advance the knowledge that we're so committed to and passionate about, and be able to, strike this balance of thinking about the future and envisioning what we think that future could be, and also being in the moment and doing that, being in that moment and succeeding in doing it well. I think it's just a skill set that we've worked on for years and years to be able and train this way to constantly think about a revised daily world. And that's at least helped me in the past several months.
Yeah, that's good.
Just to throw out another idea, something that comes up often in our Catalyst conversations. As many of you know, we've been doing this for about 8 years and have been talking to many scientists and many artists of all kinds. Anyway, is the parallel in the process of exploring and manifesting ideas. I think we've touched upon that a little bit for both artists and scientists. And of course, the outcomes are very different. And Cigall, maybe you could keep discussing sort of, or share with us the process in the research setting.
Sure.
Yeah.
Sure. I constantly use one analogy or metaphor in describing what we do, and it relates to how we run a lab on a day-to-day, person-to-person level, and also I think it strikes a chord with how we think about the science. And that is, I have a picture in my office, not my home office, but my real office, of Michael Tilson Thomas, the conductor of the San Francisco Symphony for many years. And it's a picture of his back, the leader of the symphony, with his hands up, conducting, and the entire orchestra in front of him. And for me, I always look at that as an inspiration for how I run the lab. But really, it's showing his back, and it's showing the face of everybody who's working so hard to produce all the results that we have. And it's a symbol really for how critical it is that everybody is in sync. Everyone's in harmony with one another. Everyone is in the right, is striking the right chord at the right time. And similarly, as I described to you, our goal is understanding gene expression, how genes are turned on and turned off at the right times. They have to be in harmony, in tune with one another. Just one chord that's off can cause disastrous consequences for the cell, for the tissue, and for the entire body. And so I think that metaphor really of a symphony that's working well, a symphony in which the musicians are highly in tune with one another, with the conductor, with their instruments. Similarly, the scientists have to be in tune with their tools that we have to execute the various experiments. And I think that really has been something for me that's I always come back to it. I can always find yet another parallel in the kind of business that we do.
That's great. Does anyone want to respond in addition about sort of how you make something, the process? Maybe Michelle, can you?
Yeah.
And actually, I just want to say it's very interesting that both of you kind of harken back to the 19th century. It's very interesting. Just speaking of heirlooms, thinking of Bina's discussion of heirlooms and brings you forward. So you're kind of channeling that. And of course, in scientific research, you're always channeling, 100 years back, forward, and so you're part of that that larger conversation. So.
That's you said it beautifully. That's just what I was going to bring up. I mean, I love that piece in your book, Bina, about heirloom keeping, and I do think a lot about that historically because I'm referencing other scientists and artists who have been involved in sort of translating scientific information. That idea of being very specific to that work and translating it or bringing it into a contemporary context, but at the same time honoring those scientists and artists is important to me. And it is, I think, in a way, a kind of heirloom keeping. And hopefully that's something that we then can move into the future. I think about that also in terms of my teaching. The information that we share that hopefully will be shared, with a younger, in my case, a younger generation who can then move that forward. So I think that there are so many overlays, and I really did appreciate that notion of heirloom keeping that you described. Yeah.
Maybe we'll, I want to make sure the audience gets a chance to ask questions. So maybe this will be the last thing that I pose, which is, and I've noticed this way before the pandemic, of a kind of paradigm shift in the art world, especially. Anyway, the question is, for both scientists, especially, and artists, there's a strong desire to fix or heal in this moment. And the question is, how does that play out now in your current projects? It could be for anybody. I mean, I think Michelle, you touched on this, that you're not just like, I mean, what you're doing has has a lot of sort of depth. It's not just what we see,
Right.
It's what's behind. It's what the thinking is behind what we see.
Absolutely. Yeah, I mean, that's critical, I think, to what I do. I'm interested in aesthetics and pulling the viewer in and then sharing the information, the conceptual underpinnings of the work. And I think that's what an artist can do, is bring the viewer in. And I think, Cigall, you were talking about that in your work too. So bringing them in to then reflect on the science and then coming deeper into an understanding of what that all means. Yeah.
Yeah, I think this time has been, in our field, a clear, stark reminder of the importance of human health. And so, and the importance of the science. Our lab is a cancer research lab predominantly, but we study anything that involves the process I described of just understanding how our genome is regulated, how the architecture is controlled. And it turns out that even in COVID, there are issues exactly where their underpinning is down to this process that I'm describing of gene regulation. And so I think it's, if anything, intensified our commitment to this very broad area of science that relates to so much beyond what we had, set out and charted as a path for ourselves in the lab. And that has contributed and fueled this desire to heal. It's contributed to everybody's interest in the work. It's contributed to the fury and vigor with which we approach our work, and the commitment to overcome the obstacles, including those that are current obstacles, to doing the kind of work that we had thought we would have expected for the rest of this year before the pandemic hit. So.
Does anyone else want to respond to that? Bina, do you have any thoughts?
I think maybe more so than maybe poets. I mean, I write a little bit of poetry, but I certainly wouldn't hold it up to any of the poets I most admire. But I think I come to my work from a healing place. Yeah, I think about the world's problems in a way that drives a great deal of my work, whether, and I used to teach, I'm not teaching at the moment. It's not the case that every piece of writing necessarily tries to solve a problem or heal a wound explicitly or intentionally, but I think most of the animating circumstances are the problems, really the health of people on the planet, and a sense of love that sort of drives that, a sense of love for this world and nature, and this world for people, for human beings, and hoping that they thrive.
Yeah, that's absolutely terrific, and I love to hear the word love in these kind of conversations, because I think that's for many of us, that is, we don't talk about it too much, but that certainly is an underpinning. And I just want to mention, we do not have any questions, so we can go. We have about 4 more minutes. And maybe, well, I wanted to end with this, but I'm going to say it now. It's a quote from the coda, of the Optimist telescope. And these are Bina's words. My own source of hope derives from having learned that even in this reckless age, we are not powerless. What I once believed was inevitable about human nature is rather the result of decisions we have made in the past. How we now use our power to shape the future is a choice. So I think that sums up what we've been talking about here. And, just on a personal note, it has given me, reading the book and just thinking about these things deeply has, kind of energized me to think, there are many of us thinking about the future in a positive and useful way. And that's what we need to keep doing this. And, this week, it's interesting. We are a week after the election, and there's still so much going on. That's putting it mildly. But anyway, I think we're in good company here. So maybe we'll end with that, unless anyone has. Does anyone have a coda to the coda? Well, Bina, did you want to say one more thing?
I was just going to say I'm so grateful that that you all engaged with the book this way, and I'm grateful to be engaged with this group of people, and just whether it is about the future or the eternal present. I think our relationship to time, our relationship to human life, and what it means to be alive right now is a wonderful thing to contemplate with all of you. So thank you.
Yes, thank you for the opportunity.
Yeah, I think that we'll end it there. We are almost at 2 o'clock, and I just want to say again, thank you to all of you for sharing your work and your thoughts, and for a great and important conversation. It feels like we could do another one of these. And thanks to the audience for attending. It was great to have you. I just want to mention the last Catalyst Conversations program for the fall will be December 2nd with media artist Brian Knapp, who was last with us for a program in 2018 with the architect Andrew Witt. I encourage you to listen to our podcast, Listening to Catalyst. We had a wonderful talk with Deborah Wise and Lee McCasker Gardner of the Central Square Theater. They do art and science and theater. And so that was connected to our previous 2019 Women in Art and Science. So again, thank you, everybody, and let's stay connected.
Thank you.
Thank you so much. It's great to be with all of you.